Hi everyone. Today topic about Yersinia pesticides. The Yersinia pesticides nothing about Bacillus bacterium cause a plague. Airlized bacteria can use a bio weapon also. The disease is a zoonotic disease. Yersinia, the family Enterobacteriaceae, genus Yersinia, the species of three important medical importance: Yersinia pestis, Yersinia enterocolytica, Yersinia pseudotuberculosis. Next one, general characteristics: Coccobacillary, ovoid or rod-shaped, bipolar staining. Yersinia pesticides, gram-negative bacteria, non-motile bacteria, aerobic or facultatively anaerobic, grow on ordinary media. Optimum temperature 20 to 30 degree Celsius. Biochemical test: oxidase negative, catalase positive, indole negative, ONPG, full form orthonitrophenyl beta galactoside, is positive, identified by the enzyme test ONPG. Urease positive except Yersinia. Next one, cultural characteristics: polymorphism in old culture, capsulated form in exudate from the lesions, in broth, hanging growth, seen with oil overlay, the granular deposit if it no oils. The colonies on McConaughey agar disappear after 2 to 3 days due to autolysis. In this slide, you seen about Yersinia pestis bacteria microscopic weaving under the oil immersion. Next, antigenic structure: lipopolysaccharide. Next one, capsular envelope antigenic complex. In this F1 antigen, heat liable, develop at 37 degrees Celsius. Next, stomatic antigenic complex. In this V and W antigen, heat stable, develop at 20 degrees Celsius and the 37 degree Celsius. Next one, antigenic structure to other virulence factor are low calcium response. Low calcium response decrease calcium level in a human body after affected with Yersinia pesticides. Next one, Yolks. Yolks is Yersinia outer protein. This outer protein have a 6 protein. First one, Yolks E, Yolks M, Yolks O, Yolks T, Yolks P, Yolks H. These 6 proteins are useful to human beings. Next one, coagulase produced at 28 degrees Celsius. Coagulase test nothing about mixing the bacterial cell into the large volume of the plasma in a small test tubes. When clot formation are positive, no clot formation the negative. Another plasminogen activator is dissolving the blood clot. Dissolving the blood clot in a human being that block blood flow to the brains. பிரெயினுக்கு சர்க்குலேட் ஆகிற பிளட் வந்து பிளாக் ஆகிடும். திஸ் பிளாஸ்மோஜன் ஆக்டிவேட்டர். திஸ் 4 ஆர் virulence factor of Yersinia pesticides. Next, seen about pathogenesis. The plague infected in human and other mammals. How to transmission: wild rodent epizootic. First, you know epizootic. Epizootic is nothing about outbreak of the disease. This opposite enzootic. Enzootic, low level of the outbreak the disease. Okay, first, the wild rodent epizootic means wild animals. The normal animals eat with infected animal, the disease will spread. After the disease spread to the semi domestic rodent. Semi domestic rodent example like rat, mice, guinea pig, squirrel, that semi domestic rodent that infected. Next, domestic rodent, you know, domestic rodent. After the domestic rodent to flag. Flags means insect. Next, insect to transmitted to man. The man transmission. Insect after and airborne. Pneumonic. Next one, the injection into the man by the insect. The regional lymph nodes, blood streams, organ, spleen, liver, kidney, others. Causes a infrax, necrosis, hemorrhage. Infrax, tissues damage, necrosis. In this human blood flow affected by the virulence microorganisms. Hemorrhage, you know about hemorrhage. Next one, clinical types. In this clinical types, three types: bubionic, septicemic, pneumonic. Bubionic, it's a dangerous plague infected in human. The incubation hours is 2 to 8 days. In the incubation days, septicemic. Septicemic is in this blood present the Yersinia pesticides. The incubation hour within a 1 day or 2 day, the exposure. Last, pneumonic is affected in lungs. The incubation hour 1 to 3 days. Others, plague meningitis, tonsillus, plague cormon. Laboratory diagnosis specimens: bubo, aspirate, sputum, blood, other depending on clinical presentation. Next, test staining. As usual, gram staining, methylene blue via sun staining. Next, cultural serology. Serology is ELISA, immunoelectrosporosis. Epidemiology, the disease of antiquity. In the first document epidemic 1320 BC. Next, in the major pandemic, Justin Plague, 6th centuries, Black Death, 14th centuries, Oriental Plague, late 19th century. Epidemiology too, primarily of the disease, rodent and the insect. The case of fatality if untreated, 50 to 70% of bubonic plague and 100% septicemic plague. The incident declined globally since early 20th centuries. Some countries continue to be affected. Northeast parts reservoir, the rodent and the insect plague. The worldwide disease spread between a 15-degree north, 35-degree south, Tazina central, north and the eastern part. In this slide, you seen about the world distribution of the plague 1998. Like yellow color countries reported plague 1970 to 1998. Red color you seen the region where plague occur in animals. Epidemiology 3. Epidemic types: rural plague and the urban plague. Risk factor: occupational. Farmers are many affected in this disease. Poor housings. House will be clean and the neat. Sleeping on the floors, not keeping cat. Socio-cultural factor. Next one, control and the prevention. Control of epidemic: mitigate public hysteria, treat cases, closely monitor contact, investigate all death, kill insecticides, kill rodenticides. In this slide, the case recognition, medical intervention, and the field of investigation. Identify the source of infection. Next, control the infection. Third one, the plague transmission could be seen about and the stop the transmission. Verify that patient have been placed appropriate antibiotic treatment and the local supplies of antibiotic should be handle the cases. Isolate pneumonic plague patient. In this disease, vaccines are not available still now. The continuous surveillance under the rodent and human plague. Conduct the investigation to identify the animal insect species implicated into the plague. Enzoonotic low level of the outbreak the disease. Next, the develop the program environmental management to limit the potential spread. Next, active long-term surveillance of the zoonotic. Zoonotic means animal to human disease transmission. The rapid response to the reduced exposure during episodic outbreak to control the episodic outbreak. That's it. This topic is over. Thank you, everyone.